Jai Sri Ram, Jai Sri Ram, Jai Sri Ram. In this video, we will discuss what is bacterial spore or endospore. So, what is bacterial spore, students? See, this bacterial spore you say or you call it endospore. What is this endospores? Like we have one bacteria, suppose this bacteria which is prokaryotic cell which have cell wall, and then it have plasma membrane also, and it have cytoplasm also, and then they have circular DNA and ribosomes present. Cell organelles are not present. This is a normal bacteria, this is a general bacteria. Whenever bacteria go through the kind of unfavorable condition, in that unfavorable condition, this bacteria, for surviving, for saving themselves, for saving their existence, for protecting their genetic material, they will form one dormant stage within the cell. Yeah, dormant still they will, the dormant cell, some kind of resistant stage they will form within that same bacterium in the unfavorable condition, and this dormant stage is only called endospore. Yeah, this dormant stage is called endospore. So what is endospore? Endospore is the dormant stage of the some bacterium, which not in every bacteria, in some bacterium which allow to survive in unfavorable condition. Yes, so it's very clear that endospore is going to form in the unfavorable condition. Now, the bacteria, not all bacteria will form this endospore. The bacteria like Bacillus thuringiensis, Clostridium tetani, Clostridium perfringens, these all kind of bacteria will form endospore. And maybe you have heard this Bacillus thuringiensis bacteria is used, yeah, it's used for, it was used in the biological war kind of thing also, yes. So endospore, is the resistant endospore. Whenever endospore will form, so it will protect bacteria from the drought, water scarcity. Whenever bacteria will face water scarcity, then that bacteria, will form endospore. Or low nutrient condition is there, then also this bacteria will form endospore. Or bacteria is facing some kind of radiation, they kill the bacteria like UV radiation, they damage the DNA. In that condition also endospore will form. And then temperature also, high temperature also will cause bacteria to form endospore. And the various kind of disinfectant. What is disinfectant, students? Yes, what is disinfectant? Disinfectant is actually is a chemical substance. Disinfectant is chemical substance which kill the pathogenic microorganism. Yeah, it's kill the, like in this era of the COVID, you all know this, you are using sanitizer or something. What is that sanitizer? Sanitizer have 70% alcohol, right? The what is that 70% alcohol? That 70% alcohol is only disinfectant which will kill the pathogenic microorganism. Yes. Now this endospore, this endospore is multi-layered structure. It's a multi-shelled structure, dehydrated structure. Water percentage, is less here, right? And because of less water presence, and because of multi-layered structure only, it's going to protect the existence of bacteria. So it's a kind of, it's a suspended condition of bacteria. Bacteria is not died, bacteria is live only, but this bacteria, is present in the suspended condition. And this endospore, it's contain complete copy of the chromosome. They have chromosome, they have the circular DNA and chromosome present, and the all essential proteins which is required for survival of this endosperm, they are present. And one unique thing you'll observe, they have lot of calcium ion. Yeah, the percentage of calcium ion in the cytoplasm is high in the case of endospore formation. And then they have lot of dipicolinic acid. What is this dipicolinic acid? This dipicolinic acid, is a kind of amino acid which is formed as an intermediate during lysine biosynthesis. Yes, when lysine amino acid will form, so during lysine biosynthesis, this dipicolinic acid will form. Fine. So this is the spore, you see. So many layers are present. You see, it have exosporium, then they have spore coat, and then they have core, and the inside one viable cell present. Outer membrane also many things are present. We will see one by one. And this is the bacteria having spore, and this is stained bacteria actually. We are using malachite green and safranin for the staining of the bacteria. We will see in detail. This endospore, is make bacteria to survive for a long period of time until environmental conditions are not getting favorable. Once the environmental condition will get favorable, the pH, the temperature, the exposure of radiation, availability of nutrients all will normalize. Again this endospore, it will germinate, and one vegetative bacteria, one normal bacteria will develop. Remember student, this endospore formation, is not any kind of reproduction process. No, this is not kind of any kind of reproduction process. Why reproduction process? In reproduction process, you all know, one organism will form many organisms, right? But it is fixed in one endospore, only one bacteria will develop. Fine. So endospore, what it do? As we discuss, endospore will make bacteria survive in unfavorable condition. See, the main thing, is the bacteria ultimately formed for protection of the genome of the bacteria. Yeah, the spores form within the cell, and they are full copy of bacterial genome. The endosperm not able to reproduction. Yeah, we discussed it already. Only one new cell germinate from the each spore. The spore can variable in size and location within the cell. Yes, this is important point. The spore which will form in the bacteria, it's possible the spore will be present in the center. This possible spore will be present in the terminal. This possible spore will be present in the subterminal. We will see in the next slide. Fine. If we will compare one vegetative cell and endospore, endospore is suspended cell only, and if you are going to compare this vegetative cell and endospore, what will be similarity and differences? Let's see. If you see this dipicolinic acid is amino acid which form as intermediate of lysine amino acid. In vegetative cell, this dipicolinic acid is absent, but in endospore it's present. Yeah, in endospore this dipicolinic acid is present. Fine. This is one thing. Second thing, if you'll see enzyme activity, obviously enzyme activity is very high here, but in the endospore enzyme activity is low. Messenger RNA, lot of messenger RNA will form because they have to form lot of protein. But here, in endospore, messenger RNA formation will be very limited, or it will be absent, because the cell is present in the suspended condition, and heat resistance here bacteria vegetative cell have low, and it has very high. Why? Because of presence of calcium dipicolinic acid, right? And radiation resistance is also very low here, and radiation resistance is high here, right? Water content is more here. The requirement of water is more. In the vegetative cell, they have 80 to 90% of the water, and this is 10 to 20% of the water. Endospore has 10 to 20% of the water. Now pH, you will see they have neutral pH. Fine. And the endospore have 5.5 to 6.0 pH, right? Calcium content is low here, calcium constant is high here. Fine. So this is what, vegetative cell and endospore. Fine. Okay. Now we will see how this endospore form. Oh my god. Okay, now we will see how endospore form, but before going there, let's see the how bacteria, what is the structure of this bacterial endospore. So as we discussed that bacterial endospore is multi-layered structure. Yeah, it's a multi-layered structure, and you see here, this is the this is the outermost layer, and this outermost layer is called exosporium. This exosporium, is the thin delicate layer is formed by protein, right? And then they have next layer that is called spore coat. This spore coat, is present below the exosporium and is formed by different kind of protein, so it's also proteinaceous layer. And then they have a thick layer. This thick layer is called cortex, and this cortex, is made by peptidoglycan layer. Yeah, peptidoglycan, if you remember, peptidoglycan is a substance which forming cell wall of the bacteria. Fine. So the cortex is formed by peptidoglycan. It's a thick layer, right? And it will be too sensitive to lysozyme. Somewhere this sensitivity to the lysozyme, it will help during uh, veg the germination of this spore whenever required. So cortex is a thick layer made by peptidoglycan, and then innermost layer is the spore cell wall or core wall, which is present below the cortex and it's surrounding the spore core. Okay. And what is spore core? The spore core is the normal cell which have all minimum things required for the cell, right? They have cytoplasm also, obviously cytoplasm volume is less, and they have ribosome also for protein synthesis. They have nucleoid, means DNA also present, and they are metabolically almost inactive. Metabolic activity is very low here, right? Now it is observed that in the spore core of the bacteria, germ that energy for germination, is not stored in the form of ATP. It's stored as 3-phosphoglyceric acid or 3-phosphoglycerate. Yeah, it's a great difference. You see, energy is required for germination of spore or not, so that will actually form in the form is stored in the form of 3-phosphoglycerate or 3-phosphoglyceric acid. This endospore contain large amount of dipicolinic acid, and it's complexed with the calcium ion, which making this endospore highly resistant to heat. Yeah, highly resistant to heat. Yes. Now question is this: how will we stain it? How will we color it? What is staining? Scientific method of coloring the things. Cell are transparent kind of things. It's not possible to see the cell until unless you will stain them in the microscope, right? So we are using one process that is called Safer-Fulton procedure. Safer-Fulton's procedure, the so interesting. Here, what we will do, we will take one slide, and on this slide, spore forming bacteria we will take. Okay, and we will heat fix them here. After heat fixing them, we will cover them with the malachite green, which is primary stain. This malachite green, because this malachite green, what will happen, it will enter inside the spore, because once you heat fix it and again you heat it lightly, so when you heat it, so they will enter inside the spore and they will become green color. Okay, after that we will wash it water. Okay, after washing with the water, actually when you use this malachite green, so either it is endospore or it is bacterial cell, both became, green color. But when you'll wash it, when you'll wash it, what will happen, the spore, will lose its color. Okay, spore will lose its color, and then we will use sorry, bacteria will lose its color. Bacterial vegetative cell will lose its color, and when bacterial vegetative cell will lose its color, now then we will use counter stain, that is safranin, and the safranin will make the bacterial color red color and the spore will remain green color. I am repeating again, try to understand it. See, I am using Safer-Fulton procedure. In this Safer-Fulton procedure, what happened? This is one bacteria, right? And in this bacteria, I am using malachite green and I am heating it. Because of heat, this all malachite green will enter inside this bacteria. Yeah, it's enter and everything gone green color. Okay, everything gone green color. Yes. After that, so see here everything on green color, either bacteria or bacterial endospore, right? Then what you did, you washed it, right? When you washed it, the spore remain actually green color, okay, and remaining part of the bacteria they lose the color. But how will we know it? After that we will use one red color stain that is called safranin. Yes. So after using the safranin, the bacterial spore which was green color, where the things were entered because of heating, where the stain was malachite green was entered because of heating, that will remain green color. But remaining part, they will change into the safranin color. Now what is color of safranin, student? Red color. Yes. So this will be appearance of bacteria. Now the vegetative part of the bacteria red color, and the spore is green color. Fine. So this is my uh, our Safer Fulton procedure for the study of the spore. Now question is that how spore formation occurs, right? So spore formation, so interesting. Whenever the bacteria faces is the starvation kind of condition, this process of sporulation begin. Okay. The first change we will see, what first change we will see, that within that particular bacteria, the DNA will replicate. Okay, and DNA will replicate, and two copy of DNA will form. After that, what happened? The first change occurred during the spore formation. Both parent and daughter chromosome, they will arranged into the axial filament. Try to understand it. Like I have one bacteria, you see here. See here, this is the bacteria, right? They replicated the DNA, two copy formed, right? And now they will arrange at the two poles kind of thing. We are calling it the axial filament. This both DNA will arrange. And then what happened? Then yeah, then spore will start to form, right? And then they will, the spore, you see spore is dividing, the septum kind of thing will form, spore will divide, and after that possibly the bacteria will die also, right? Spore will survive. Whenever again favorable condition will come, the bacteria, the spore will germinate. This is the spore, it will germinate, and the vegetative bacteria will come. Fine. Okay. Now, what are the examples of the spore forming bacteria? We all know dangerous bacteria, very severe kind of food poisoning they causes, Clostridium botulinum causes food poisoning, botulism, very dangerous, many times causes death even, right? Gas gangrene, oh my god, gas gangrene caused by Clostridium perfringens, very dangerous disease, which only surgery will help here, then Clostridium tetani, that causes tetanus, and the Bacillus anthracis is also spore forming bacteria that is causes anthrax. We are using as a biological weapon also, right? This is how, the after staining, that suffer Fulton procedure, this is how it happened. The part of bacteria because of safranin red color, and the spore will be the green color. Fine. Remaining we'll see in next video.